Hello, everyone. Hi, it's Long.
Aaron.
How are you doing?
Action. Aaron.
Hey.
Yeah.
Hi, Viviana.
I'm still in the hotel, so I'm trying to see the connection is good.
Hi.
Seems good. Hi, Viviana. Thanks for coming. Can you test out your slides?
Sure.
Is he coming yet?
He sent me a note that he's trying. I don't know what that means.
Oh, great.
Looks great. Nice. Okay. Thank you. So I'll just have a quick introduction, Viviana, about the meeting and then of you, and then you can speak first.
Sounds good. And you can hear me all right?
Yeah, great.
Good.
Perfect. Okay.
How is going?
Good. Good to see you.
Figure out how to use Zoom.
Yeah, it's with the periodic updates, I find myself being late for meetings. Like I click to log in and then they say you need the next version of Zoom without any heads up when that might be. So I'm glad you. Okay.
Yeah, we usually try to get the speakers to come in about 10 minutes early, just in case there's any problem.
Hello?
Yeah, I hear you. Great.
Is it possible for me to go first because I have a meeting coming up after this?
Sounds good.
Okay.
No problem.
But I
Works for
need
me.
to test whether I can do it. I've never done this before. So I need to do
Share
the
screen.
sharing.
This is your first Zoom meeting?
Ever. I've
Really?
listened to Zoom meetings, but I've never done one by myself.
You have to share your secret.
I wish I could never have done a Zoom meeting.
And then
open a PowerPoint and share your screen. Point to the PPT you want to share.
Oh, okay.
Yeah, here we go. Here
Is
we
it
go.
on?
Yeah,
And
great,
then I
great.
put it full screen.
Yeah.
Oh,
Here we go.
okay, that's not so hard.
Congratulations. Excited to be your East first Zoom meeting.
Right, my first ever.
Hopefully it'll be a good experience for you.
Cool.
How do I? Oh, I see. Take the voice.
And then you could just stop sharing the screen for now, and then you could share it once you're up.
And how, stop share here. Okay.
Great.
Thank you.
You
Is it okay?
Figure out, right? Bailu come to the Zoom meeting, figure out where. So congratulations, you get new technique.
Is it okay if I change your name on here to your name, just so people know it's you?
Sure.
Sure.
Yeah.
Thanks. Thanks. And then let me make you the co-host. Okay. Great. And then in about 1 minute, we'll.
And do I answer the question after my talk or at the end?
After your talk.
Yeah. Try to go no more than 25 minutes, and people have a discussion time.
Now, how do I have a clock? Oh. Reset. I'm setting a clock for myself here.
Nice.
Okay.
So I can see.
Okay, maybe Zilong, should we get started?
Sure, yeah.
Okay, so thanks everyone for tuning in to what will be an exciting Neuro Zoom session. And just as a reminder, next week we have more talks coming. We have Juan Loa from Peking University and Yadong Huang from UCSF. So please tune in for those and let me or Zilong know if you'd like to present your work here. With that, we'll jump right into Zilong introducing the first speaker, Yiro.
Okay, great. Thanks, Aaron. So thanks for everybody for coming. So I'm very excited to have introduced the long-term, not only friend, also mentor of mine. So I'm gonna give a very short introduction for Professor Yi Rao. So Yi has graduated from Jiangxi Medical College, a bachelor degree, and after a short time period in Shanghai Medical College, but without a master degree, he went to UCSF. Worked with Yudong Jian and Li Jian, to start his PhD in neurodevelopment ever since. So after PhD, he went to Dr. Melton's lab in Harvard. Will continue working on neurodevelopment. So after a postdoc, he started his facultyship in the Washington University, where he started his journey to discovering amazing series of slit, roll, guided molecules. After Washington University, he went to a short period on the Northwestern. But most importantly, he went back to China in 2007 to become a dean of the School of Medicine, a life science school in Peking University. So he's continued his work in China in Peking University, as well as now become one of the chancellor in another capital medical university. So in his own lab right now, you can find out he also has wider interest, not only neurodevelopment, but also on a screening about the mechanism, genetic basis for sleep. I believe he'll talk about today, also as well as several other interesting aspects. Okay, today we'll come around to share with us his latest discovery about the interesting molecule regulating sleep. Please take it away. All right.
Hello, this is my first ever Zoom meeting that I am the speaker. For the last few years, my lab has been studying mechanisms of sleep. We've published a few papers on neurotransmitters and receptors involved in Drosophila sleep. We've also corrected a debate in mice whether serotonin is involved in sleep regulation or not. And our conclusion was, yes, serotonin is indeed required for sleep in mice. What I'll be talking about today is a very specific question on the molecular mechanism of sleep need. According to Alexander Borbeli, there are two processes that regulate sleep. One is the process C, which is the circadian clock, as we know. The other is process S, a mechanism that we know much less about. What my lab has made a discovery on is on sleep need. And sleep need is very important for sleep. It drives sleep, and it is itself homeostatically regulated. That is, when you have more sleep, you need less. When you sleep less, you need more. And for example, if you studied too hard last night, then you will be compensating for that. You have a recovery after sleep deprivation. So far, there has been studies on what might be molecular or cellular control of sleep. There are also attempts to look at what might indicate sleep need. But no molecule or cell satisfies all the criteria for being both an indicator and a controller of sleep. Masashi Yanagisawa in 2016 published a breakthrough paper on salt-inducible kinase 3, SIK3. What his group discovered was a mutant that delete a segment of the SIK3 kinase. And this deletion included a site, serine 551, which is a target site for PK phosphorylation. With this deletion of either S551 alone, which they did later, or the segment covering this site, which they published in 2016, they had a gain of function mutant for SIK3. That is, the function of SIK3 was increased. And when this was increased, they got a mouse mutant, sleepy, that sleeps more. So with an increased function of SIK3, you have more sleep. And this is a major finding on the molecular basis for sleep. Now, later on, Yanagisawa's group have published papers on SIK1 and SIK2. In all these three SIK kinases, if you mutate the S551 site, you get a gain function in SIK1, 2, or 3. And when they have gain of function, you have more sleep. So, and you have more sleep need. Now, this is an interesting finding in and out of itself. But because they are all gain of function mutants, there is no evidence for loss of function mutants that would indicate they are required for sleep. Furthermore, SIK1, 2, 3 phosphorylation on 551 does not correlate with sleep need. It actually does not change over 24 hours. So we went on to study SIK1, 2, and 3 by generating loss of function mutants. When we knock out SIK1, as you can see here on the top left, that's mRNA level of SIK1 homozygous wild type, heterozygous, and homozygous mutant. You can see there's a dosage-dependent decrease of mRNA of SIK1 when we do the SIK1 knockout. On the lower right, what you see is a measurement of sleep need. The commonly accepted measurement for sleep need by electroencephalography, EEG, is to look at non-rapid eye movement sleep period for delta power density. Delta power density is the 1 to 4 hertz EEG oscillation. And looking at this measure for sleep need, what you can see that there's no difference among SIK1 homozygous wild type, SIK1 heterozygous, or SIK1 homozygous loss of function mutant in sleep need, indicating why Yanagisawa's lab has published that SIK1 gain of function increased sleep need. SIK1 loss of function has no change in sleep need. So that is why you generate an artificial phenotype for SIK1 when you have gain of function. It is actually not physiologically required for sleep need. Similarly, we've generated SIK2 knockout, and you can again see a dosage-dependent decrease of SIK2 expression of its RNA on the upper left corner. And again, when you look at NREM delta power density as measurement of sleep need, there's no difference between SIK2 homozygous wild type, heterozygous mutant, or homozygous loss of function mutant, showing SIK2 is also not required physiologically for sleep need. By contrast, when we generate SIK3 mutant, we see a change. Now, SIK3 knockout, now phenotype, now mutants are largely lethal. We've looked for hundreds and thousands of progenies, and it is not impossible, but it's very few 6'3" null mutants can be discovered. On the other hand, we generated a 6'3" knockdown mutant, so that 6'3" is not completely wiped out, but the expression of RNA is reduced, as you can see in the upper left corner. Again, in a dosage-dependent way, in our knockdown mutant, 6'3" mRNA is decreased in heterozygous and further decreased in homozygous mutant. In the lower right corner, what you can see is that when you look at sleep need measurement by NREM delta power density, what we can find is that 6'3" homozygous loss of function mutant or knockdown mutant has a significant reduction of sleep need. And you can measure that by at different time points of ZT0, ZT2, ZT12, ZT14, 18, and you can see the sleep need reduction is significant statistically from ZT12 on most of the time. Furthermore, you can compare ZT16 with ZT8 or ZT20 with ZT8. What you can see is that with the reduction of 6'3" loss function mutant with mRNA reduction, you can compare time-wise, the time that requires to make a difference is also delayed. And this is the comparison of different ZT time. Normally by ZT16, ZT17, the sleep need would be different from ZT9 or ZT10 in the wild type. That's the top panel. At the bottom panel, you can see when a ZT6'3" knockdown mutant, that by ZT16, 17, 18, 19, there's no difference of sleep need between ZT16, 17, 18, 19 with ZT9. Only there's a slight difference, there's some difference by ZT20. And again, the difference disappears in ZT21, 22, and 23. So by all measurement of delta power density, the sleep need is significantly reduced in 6'3" knockdown mutants. Another measurement for whether a molecule is involved in sleep need is to look at sleep deprivation. So if we deprive the mice for sleep for 6 hours and then allow the mice to sleep normally, it would have a rebound. So it's a sleep rebound after deprivation. In a wild-type case, shown in black curve here, what you can see is that there's cumulative rebound after sleep deprivation. On the other hand, in either the 6'3" heterozygous knockdown or homozygous knockdown mutant, sleep rebound after deprivation is significantly different from wild type. So what we have shown is that SIG1 and SIG2, while gain of function increase sleep need and sleep, loss of function shows that they are not physiologically required for sleep need. On the other hand, if we look at the knockdown 6'3", we find that it is physiologically required for sleep need. So 6'3" as a protein or kinase controls sleep need. We further look at whether 6'3" can serve as an indicator of sleep need. 6'3" has multiple phosphorylation sites. The site mutated in the initial sleepy mutant that removes S551 is phosphorylated by pKa, but its phosphorylation does not correlate with sleep or sleep need. There's no change. We look at different site, serine 221. First, we show, by we, I mean postdoc Yuxiang Liu and technician Yunfeng Cui, showed that on the left panel, that if 6'3" is dephosphorylated by phosphatase, then its activity on its substrate, HDAC4, is reduced. Furthermore, on the panel on the right, if we mutate T21 into alanine, what you can see is the single mutation of T221A wipes out the 6'3" phosphorylation of its own substrate, HDAC, showing that T221 phosphorylation regulates or promotes the catalytic activity of 6-3 kinase. Furthermore, Yu Xiang and Yunfeng showed that when you transfect 6-3 into HCK cells and block protein synthesis by cyclohexamide, what you can see over the next 24 hours, there is some reduction of the protein over the first 2 hours, and then it's pretty stable. If we mutate T221 into A, what you can see is that over the next 24 hours after protein synthesis is inhibited, there's rapid degradation of 6-3, indicating that T221 is required for 6-3 stability. And this is also the case, not only in HCK cells on the left-hand side, but it's also in the mouse brain. When we mutate T221 to A, then the protein stability of 6-3 is also significantly reduced. We have obtained an 6-3 T221 specific phosphor-specific antibody. What we can see is in this amazing figure shown here in front of you, that over 36 continuous hours, starting with light on as ZD0, what you can see is while this level of actin is stable and the level of total PER2 protein changes in an interesting manner, the level of 6-3 T221 phosphorylation and T6-3 total protein decreases over 12 hours of sleep. When you sleep more, you have less 6-3 phosphorylation and less 6-3 protein, because the 6-3 protein stability correlates with 6-3 T221 phosphorylation. Once you start to sleep at the end of 12 hours of playing, then the more you, and once you start to play, the more you play, then the more T221 phosphorylation you get. So you increase T221 phosphorylation from CD12 to CD0. Once you start to sleep again, or once the mice start to sleep again at the second CD0, then phosphorylation starts to decrease again. So this is an amazing correlation between sleep need and the phosphorylation of a single protein at a single site. And we can also see that with sleep, in a normal sleep, ZD0 has higher T221 phosphorylation than ZD6. But if the mice has been deprived for sleep for the same 6 period, what you can see is that the endophosphorylation did not decrease. So using both a natural daily cycle to show that T221 phosphorylation correlates with sleep, and to confirm with sleep deprivation, what Yanagisawa's lab have also seen, that in sleep deprivation, you can also increase T221 phosphorylation, while you cannot increase or change the phosphorylation of other kinases such as p-ERK or p-JNK. Furthermore, when we generate 6-3 T221 mouse mutant, what you can see is a sleep need as measured by NREM delta power density is significantly decreased both in the heterozygous and furthermore in the homozygous T221A mutant. If we look at sleep rebound after deprivation, what you can see in both the heterozygous and homozygous T221A mutant, sleep rebound after deprivation is significantly reduced. So by these results, we conclude that T221 phosphorylation in 6-3 kinase is both an indicator and controller of sleep need. That its phosphorylation indicates the amount of sleep. And if you cannot phosphorylate, then you lose control of sleep need. To say that the Boberian model can be converted into a molecular model as presented here. We further look at what's upstream of T221 or 6-3. About 18 years ago, several groups have published papers showing the liver kinase B or LKB1 phosphorylates, or they can claim phosphorylate AMPK. And 6-3 is a member of the AMPK-related kinases. And so in 2004, Liz Cano's paper in EMBO was the one that shows LKB1 can phosphorylate 14 different AMPK-related kinases. Together with AMPK itself, the total was 14 at that time. What we look at is to look at LKB1 knockdown. Again, LKB1 knockout is lethal intracellular, but LKB1 knockdown can reduce both daytime and nighttime sleep intracellular. We also did a virally mediated LKB1 flux loss of function mutant in the mouse brain by viral injection into flux LKB1 mice. What we can see that our viral injection can indeed decrease the expression of LKB1 protein itself. Furthermore, when we look at the phenotype, we can see that sleep need was decreased in LKB1 knockdown mice. So we conclude that 6-3 T221 is crucial as an indicator and controller of sleep need. One possible upstream regulator is LKB1. But when we built up our biochemistry team in my lab, we found out it's a lot more complicated. That does LKB1 phosphorylate 6-3 directly, or does it only phosphorylate AMPK directly, but 6-3 may not so directly? We're working on that. And when we look at 6-3 kinase upstream, we actually found a very large number of kinases that can phosphorylate 6-3 at T221. So biochemistry is a lot more interesting and complicated than previously thought for the last 18 years. So we're still studying those both by the biochemistry and by looking at which one of those may again also be involved in sleep and sleep need. With that, I conclude my talk. The biochemical studies were led by the postdoc Liu Yuxiang, and postdoc Yang Li did some of the work, and graduate student Tao Wang. We have a very talented technical associate, Cui Yunfeng, in the lab, joining Dr. Liu, Li, and Wang. Functional-wise, the mouse sleep group was supervised by Zhou Wenxin, and we have two talented technicians, Yang Jinquan, Li Chenggang. We also have others joining the project. The work on LKB1 was mainly carried out by graduate student Liu Ziyi and helped by postdocs Jiang Nifeng and Li Chaoyi. With that, I can take questions.
Great, thanks Li for amazing talk. So it's amazing 221. So a quick question for now is: you have made the point mutation mice? Did you?
We did. It was part of the sleep need study was all done by the point mutation.
Right. But also, you're suggesting that the total level of 6-3 maybe also be down by about 50% in the point mutation mice.
Because they correlate. Is because T221 phosphorylation is required both for its catalytic activity and stability. So you would always see that. Yeah.
Okay, so I'm thinking maybe if there is some inducible, I don't know, it's difficult, right? So make point mutation, but not consistently, but inducible according to in some brain region, maybe more. Anyway,
we now enjoy viral injections a lot in the lab, thanks to other people who reminded us, especially Yulong Li, who's always catching on new technology. I was a little.
Yeah. Kang has a question.
Yes. Hi. any human mutations that could explain the different sleep needs among populations? Like Aaron never sleeps, as far as I could tell.
So far, not yet. It would be very interesting to look at human mutants, but we believe that there's multiple kinases that has similar functions.
Okay. So Robbie, do you want to ask a question yourself?
Sure. Can you hear me okay?
Yeah, sure. Great.
Okay, great. So I'm wondering how you distinguish between a change in sleep need and a change in the association of delta power with sleep need, which are not necessarily the same thing.
So the common measure of sleeping need is delta power, and so we did the sleep recovery after the deprivation studies. So those are the independent studies showing that it can be involved. Otherwise, there's no further distinction.
Yes. So the problem might be that when you change the SIC3, what you're disrupting is the association with delta power, your indicator, as opposed to a change in sleep need per se.
Right. It's possible, but the simpler explanation would be that it is directly indicating it.
Yes, I take that as a statement of faith.
Thanks. Any more questions?
Can I ask a quick question?
Sure, please.
Yeah, does people know where the SIC3 is actually expressed, and also which neurons expressing the SIC3 might be important for the sleep function?
Yanagisawa's lab has published a paper in Journal Neuroscience earlier this year to show that yes, SIC3 is required in neurons, but on the other hand, there's too many neurons that express SIC3. At this point, it's unclear. I'm assuming that Masashi Yanagisawa's lab is busily doing that.
Okay, I see. Thank you.
Okay. Sonia, do you want to ask a question yourself?
Yes, the question, beautiful talk. The question is actually similar to the question like from previous people. Basically, do you know whether it's really neuronal controlled or it's from the whole system? If it's neuronal control, which neuronal circuitry may be involved? And whether, for example, dopamine pathway is involved in this or not?
Right. No one has done the relationship with the other transmitters or other molecules. It's clearly neurons are involved, and Yanagisawa's lab have that result. Our LKB1 condition knockout, we use the neuronal driver, so that's also neuronal. So clearly neurons are involved. Whether other cells are involved or not is unclear, but we don't need other cells. All changes in neuronal cells of these genes are sufficient to cause the same phenotype.
For example, have you checked the dopamine pathway and the dopamine neural activity?
We would be more interested in checking serotonin pathway and other pathways. No, so far not yet.
Something like that. Yes. Okay. Thank you.
Most of the transmitters that has been known previously, when they knock out the phenotype are all minor. I mean, the top candidate is actually histamine, but even histamine receptor knockout, the phenotype in sleep was minor. So far the transmitter implications are far more complicated than the textbook tells you.
More questions?
Great, very elegant work, Emin here. I was wondering, maybe I have missed, is SIC3 under the control of the circadian rhythm system, or is it completely separate? Is it actually circadian rhythm will also use this pathway to drive sleep, or it's a completely separate pathway?
In one study, a different Japanese group have shown that 6-3 can phosphorylate per the control of circadian clock. And so there could be some feed forward from 6-3 to the clock pathway. In terms of whether clock controls 6-3, if we look at a natural daily cycle, we wouldn't be able to separate those. But with the sleep deprivation results, that shows sleep need and the 6-3 mutually controls each other.
Thanks. Yeah.
Okay.
Sorry, like maybe I ask a more related question. So have you checked other circadian response in different tissue on whether you start have a loss of the circadian control of the whole body, or you just they still have in the circadian rhythm this kind of like regulation, normal regulation?
We are comparing all these mutants for the circadian clock, and we'll show that slightly later. I've looked at it, the students' results, but I want to wait a little bit before I say it clearly.
Okay, Xiaohong, do you want to question yourself, please? Go ahead. We can raise a question for you, but.
Oh, sure. So I was just reading elegant work. I was reading that there was a report about another phosphorylation site of the same protein seemed to actually increase sleep need. So how do we reconcile these two sides of the same protein that it can either decrease or increase?
It would be very interesting. We don't know the other side, what roles they play. Is it because they stabilize the protein? Is it because they increase the activity of the kinase, or they increase the function of the kinase that responds to upstream kinase, or associated with other proteins? That's unclear.
So are there any sort of follow-up study with structure of this protein that may gain some insight about how it may regulate?
I'm sure that some structural biology would do this, but we are not going to bother with that.
Okay, thank you.
Okay. Right. I have a quick last comment, not question, but for the LKB1 upstream, I'm sure people already did a lot of LKB1 condition knockouts. You might check in the future further whether indeed the LKB1 mutation has the sleep problem. Tell us more about. Yeah, very interesting to see how it's going. Okay, if no more questions, let's thank Yi for a amazing talk today. Great, thanks.
That's my first Zoom talk ever.
We have more. Yeah, great. Thank you so much. Okay, and a second talk, Professor Viviana.
Thanks so much, Yi, for joining us and giving your talk. And now it's my pleasure to introduce the next speaker, Professor Viviana Gradinaro, who is a professor at Caltech. Viviana attended University of Bucharest in Romania for undergraduate, and then transferred to Caltech to finish her undergraduate degree, and then moved to Stanford for her PhD with Karl Diesroth. And at Stanford, during her PhD, she had over 15 papers as a PhD student, including first author papers in Cell Science and the Journal of Neuroscience, and many other papers in the top journals of our field. So when I first became familiar with her work in a famous paper she published in Science as a graduate student, in which she used optogenetics to, in one fell swoop, reveal the mechanism of deep brain stimulation, and also uncovered a better targeting strategy for therapy. So DBS is this miraculous therapy that works in Parkinson's disease, but we don't know how it works or why it works, and she figured both of those things out. Moreover, before her paper, optogenetics had held great promise for doing these types of things, but hers was the first demonstration of it actually working in both settings, and to work out circuit mechanisms and to improve therapies. And she also revealed strikingly different effects when stimulating even the same cell type in the brain with different frequencies, and this has clear implications for application of DBS to Parkinson's patients. She then was offered a faculty position as a graduate student right away at Caltech. This is unusual, but she's an amazing scientist, and her lab has pioneered tissue clearing strategies, including a method for clearing entire animals, whole organs, and bodies. She's developed new AAV capsids to deliver gene therapies to the central and peripheral nervous systems. These are now widely used by the neuroscience community. As an assistant professor, she was a Pew Scholar. This is the most prestigious award an assistant professor can receive. She's been awarded the Presidential Early Career Award for Scientists and Engineers. She won an NIH Director's Pioneer Award. She recently received the Vilcek Prize for Creative Promise in Biomedical Science. She's an inspirational scientist, and looking forward to hearing her latest. Go ahead, Viviana. You're still on mute.
Thank you so much. Thank you so much, Aaron. And I will try to do justice to the very hard work that this team at Caltech is pursuing. And as Aaron mentioned, I'm a neurotechnologist, and over the years, while trying to pursue basic science questions in neurodegeneration, in Parkinson in particular, I've been encountering barriers after barriers of technological nature. And this was the reason behind getting involved in work around optogenetics and tissue clearing, and most recently with my lab on gene delivery vectors. And these technologies now have matured enough that we are starting to have scientific fun and applying them to understand circuits underlying neurodegeneration, and hopefully finding ways, both in terms of pathways and delivery methods, to improve outcome for those suffering with neurological disorders. I have to mention as a COI, I think there's a drawing on the screen. I don't know if that's, okay. So
I'll take care of it.
Okay, thank you, Aaron. My trainees and I co-founded a company based on technology from Caltech with the hope that some of our work can translate into the clinic. As Aaron mentioned, my work on Parkinson has roots in circuit neuroscience, where I used optogenetic tools to try to understand how deep brain stimulation works. And that work was rewarding. It allowed also for development and improvement of optogenetic tools. However, both at the time and especially later on when I moved to Caltech and I had colleagues working on basic science problems on immunology, I realized more and more that we were looking at Parkinson's way too late and maybe too brain-centric. And before motor symptoms, as much of the audience is familiar with, there is perturbed sleep, and the prior talk was fantastic. And sleep can be a very good gauging factor for pathology to develop. So very important to understand the molecular basis of sleep and to pay attention to any disturbances that might happen. In addition to perturbed sleep, there can also be gastric dysfunction. And both of these implicate a much broader central and peripheral nervous system involvement. And this broader involvement actually points to the fact that we should look not only at the brain, but also at the body, and much earlier in the progression of the disease. So that was when we wanted to try to apply some of the tools developed over the years with collaborators, having to do with anatomical analysis and tracing, and also with functional assessment by using well-known methods, optogenetics and chemogenetics. And we wanted to apply them in the context of brain and body, but we encountered maybe a very significant challenge in that path. And the challenge had to do with delivery of genes and delivery of photons. So both for gene delivery for functional readout or functional control, and also for light delivery, because we're based on visible wavelengths, there's significant scattering in the mammalian tissue. So these were significant problems. And he pointed us to the fact that if we are to make progress, we should try to understand and maybe bring our contributions to the gene delivery area. And this work resulted in some systemic AAVs that can cross the blood-brain barrier. And I will update you on some unpublished work on that, and also improving the opsins to match that need. And importantly, and which was the goal all along, to try to use these technologies in understanding and maybe furthering research in neurodegeneration. In terms of gene delivery, to achieve safe and effective and minimally invasive delivery, we are so fortunate because we're benefiting from 5 decades of research on the adeno-associated viral vector, since its discovery in the 60s, all the way to the very exciting recent times that where we see the AAV not only improving outcome for patients, but prolonging their lifespan as well. So it's approved for spinal muscular atrophy in the United States, in Europe as well. It's also used for vision indications. However, for the brain, the challenges are significant still. And the use of the adeno-associated viral vector is maybe hampered by the fact that delivery is still limited. So this is a familiar method to us neuroscientists, where we do stereotactic surgery, deep brain insertions of needles, and spot injection. And the spot injection has a very limited distribution, and you have maybe too many copy numbers at the injection site that can be toxic, and away from the injection site, it's not enough. This is a mouse brain. And if we put it in the context of the larger a non-human primate brain, you can see how difficult it will be to get significant spread of biological relevance by using spot injection. And this was one challenge that over the years in the group and with collaborators, we tried to address by taking the approach of delivering genes from the bloodstream. However, the big barrier here is the blood-brain barrier. And we have it there for a good reason, to protect us from disease, but it also prevents research tools and therapeutics from crossing. And this is where the approach we took was, rather than a basic science approach, was an engineering one, because the biology of the BBB, although we're accumulating knowledge, is not fully understood. And the intersection between viral entry and BBB even more so. So because we don't have enough knowledge on the biology, the approach we chose to take, and that was successful, was to use directed evolution, which is a method that my colleague Frances Arnold in chemistry invented. And it's a method that can improve biologicals and chemicals with if you have a starting point that has the property that you want, but maybe it's not strong enough. So in our case was AAV9 that could weakly cross the blood-brain barrier. Then you generate diversity, you make a lot of offsprings from that library, and you can take this selection process in directed evolution. And this selection process can give you capsids with desired properties. And this worked, and it allowed us to take AAV9 from a capsid that crosses weakly in the adult. Again, in the neonate is different. Neonate has good crossing of the BBB, but in the adult mammalian brain in the mouse, you have a rather poor transduction and mainly astrocytic. So through engineering, we were able to cross the BBB in mice, and because only the capsid is the one changed, you can change the cargo to a desired one. And here, just for proof of concept, you can target different cell types in the brain with different cargo. Beyond the brain, we are able, through the same selection methods, to also develop capsids that were very effective in targeting the peripheral nervous system. And this goes back to this gut-to-brain problem that I highlighted at the beginning. And we had a project that used these vectors that we developed through directed evolution and allowed us to understand a bit more about the contribution of the gut pathology to eventual neurodegeneration in the brain. And this was led by a former postdoctoral fellow, Colin Callis, and Colin seeded the gut with preformed fibrils, and then look to see a developing pathology in the enteric nervous system, but also in the brain. And much to his surprise, what we learned was that if you do a very small amount of seeding, so modest amount of preformed fibrils in the duodenum, and this is important, in healthy young adult mice, there is some short-term gut dysfunction, but that returns to the baseline, and there's no long-term consequence in terms of the brain and motor functions. This was not the case when Colin did the exact same paradigm in 2 different cohorts. One, aged mice, and the other one, mice that were overexpressing alpha-synuclein, the Ty1 alpha-synuclein overexpressing mice. In those 2 cohorts of mice, the same procedure that didn't give results in the healthy young adults caused severe gastric dysfunction, and this gastric dysfunction was followed by progression with brain pathology. Colin wanted to understand the molecular basis of this and looked at a broad panel of molecular targets. And I'm showing you one of the findings where Colin saw that there was a correlation between the GCase level. GCase is a lysosomal enzyme encoded by GBA, and importantly, GBA mutations are responsible for Gaucher disease, and it's also a risk factor for Parkinson's. So what Colin saw was that in wild type, you have higher level of GCase than in the aged animals and in the alpha-synuclein overexpressors. And this is inversely correlated with the developing pathology when there was gut dysfunction induced by the peripheral fibrils. And of course, this generated the hypothesis that if we put back GCase by gene delivery of GBA, this will rescue the phenotype. So Colin used a system we developed with the systemic vectors for a dual vector system for fast expression of GCase because we were fighting against the timelines of induced pathology. We had to intervene early enough and fast enough. And you can see here the rescue of GCase in the alpha-synuclein overexpressing mice. So you have wild type level, you have the ASO mice, and then you have the GCase level upon gene delivery. And then you can also see the pathology. So this is the phosphorylated alpha-synuclein, that's a marker of pathology. And you can see how high level of GCase, there's very little, if any, phosphorylated alpha-synuclein. In ASO's high level, but you see partial rescue with gene delivery. And this molecular rescue was also followed by physiological rescue, and Colin here and collaborators in the Masmanian lab did a series of gastric of motility assessments. As neuroscientists, we were interested to see whether we can catch earlier markers of degeneration by looking at the physiology of the enteric nervous system. And this, unlike the brain, is being rather difficult to both measure and control gastric function, the enteric nervous system. And Colin generated this in vitro prep where we could do calcium imaging and optogenetic control of the gut tissue. And he was able, by using a similar multi-vector approach, to stimulate the gastric system with opsins partially delivered and measure the outcome with calcium indicators. And then he was able to show that by seeding the peripheral fibers, you can get pathology, and this can be rescued at the physiological level with gene delivery. Of course, this is just one piece of data, as is the case in research. These are very difficult problems, and this can be taken to many different areas. We are focusing now on the thing that I think we're best positioned to solve, which is trying to take this towards the larger brains. And the bottlenecks are that AAVs, whether they're systemic or not, are not specific, so they can cause side effects and confound experimental results. If you think about the dopaminergic system, if you deliver systemically to the brain, you might also get heart modulation and confound results. The PNS is still in need of better tools. And importantly, AAVs perform differently across species. So we are working on this, and I'll show you some unpublished data around each one of this and some disease proof of concept applications, again, early and unpublished. The AAVs are not specific, and we know this from published work and also work we did with our collaborator, Kathy Ferrara, that runs a PET imaging lab at Stanford, and we were able to chemically label the AAV capsids engineered and look at the distribution real time in vivo, in living mice. And what you can see very quickly is AAV9 accumulation in the brain is very modest, engineered vectors high, but this you still see it in areas like the liver. And this can be a significant problem for toxicity, and many clinical trials have been stopped or complicated by liver toxicity. So then we went to our directed evolution pipeline and we asked more of it. We asked not only to get vectors across the blood-brain barrier, but by doing multiplexed selections in Cree transgenics of different cell types and organ identities, or in terms of where Cree was present, we asked of these selections to get viruses not only that are strong in the brain, but viruses that might be weak in the liver or weak in the DRGs. And I'm very happy to tell you that this approach, the iterative sequential approach, has been successful. And we are able now to take the parent from the AAV9 parent, from a parent that's weak in the adult mammalian brain with systemic delivery, but high in the liver, to an intermediate that we published a few years ago, PHPEB, that's widely used now. And I'm very glad to see basic science research that uses this vector. It's worth noting that the brain expression is very strong, but liver is still present. To variant that's now high in the brain, and we can hardly detect anything in the liver. So this approach is powerful in terms of generating the vectors of need. And as I mentioned, for the peripheral nervous system, the tools, if anything, lag a bit behind the central nervous system. And that's why we have been advancing the variants for the PNS as well. And we have vectors that are stronger now in the PNS with, importantly, the same feature of liver detargeting. I'm mentioning these two vectors, so the ones that are detargeted from the liver, but strong in the brain, strong in the PNS, because very recently we are able to do a combination gene delivery by using a brain capsid and a PNS capsid to application in neurodegeneration for Friedreich's ataxia. It's a rare neurodegenerative disease. It's a loss of function, and it's a good candidate for gene therapy. And the key element, the key molecular element here is the frataxin protein. Not enough frataxin protein that causes a lot of complications for the children that suffer from this disorder. So my graduate student in the lab, Akeisha Hori, MD PhD student, she did gene delivery, a combination gene delivery into animal models of Friedreich's ataxia. And I'm going to show you very exciting, again, early data, but Akeisha did run enough animals for me to have confidence to show you this early data. And I want you to note, so this is a narrowing beam assay, and you can see the wild type. I hope you see it. It doesn't play as well on the transmission is good enough. Let me see. Okay, I hope you can see it now. So the wild type should move across the beam very smoothly, while the control, so this is the diseased ataxic animal, has significant slips from the beam. And when frataxin is delivered to both the central and the peripheral nervous system, you see this, the videos might be too large for good rendering, unfortunately. I'll click on them a few times and maybe they play. But regardless, we have the quantification for this. So this will let me, yes. So you're also able to see the quantification in the wild type versus the control versus the ataxic animal. And we think that this combination can rescue phenotypes for Friedreich's ataxia, and we are looking at the molecular rescue as well. Importantly, all of these capsids I presented are available on Agene, and there is a decision tree in terms of what would be best for the application. And most recently, we have crossed the challenge of delivering genes to the marmoset brain. And this is work that was led by very hardworking and a collaborative work from David Gortz and Nicholas Fleitzanes and Nick Godin in the lab, alumni, Fleitzanes, Godin, and David Gortz, and a graduate student in the lab. And what you see here is this is the marmoset brain. It's the marmoset brain, spinal cord, and you can see the distribution throughout the brain. And we're excited about this. We hope the marmoset labs, we don't have marmosets in the lab. This work was only possible because of amazing collaborators all over the world. And you will find out more about it on Thursday when it will be out from collaborating labs as well. And we hope that this kind of vectors and the ones that we are also advancing for cell type specificity can be held for the study and repair of the nervous system. And I have to emphasize it's important to do this for all brains because there's enough diversity, as we know from strains of mice. And this summarizes the key points I mentioned. We are focusing now, for those interested, for those listening and interested, I want to mention 2 efforts. Bioengineering efforts are around maximizing payloads, so to obtain tropic capsids, minimize gene regulatory elements, and AAV size increase. And basic research efforts are around the gut-brain relationship in relation to alpha-synuclein perturbation. And we have a very large team effort. These are amazing collaborators, many of which we've been working for many years and published for many years. And we are trying to take tools that have been used successfully in the brain and try to apply them to the peripheral nervous system and understand the gut to brain in the context of Parkinson. If any of the trainees on the call are interested, please email me afterwards about this effort. So this closes, so we have enough time for questions. So I've mentioned many of the trainees' contribution throughout the talk. I want to highlight alumni from the lab that have started their labs and are recruiting, some working on neuroengineering problems, some working on basic science, and also alumni that moved on to next steps, whether in academia or industry. This would have not been possible without the BRAIN Initiative. The BRAIN Initiative was the one that supported a neuroscience lab that has never done AAV engineering. We were eager AAV users, but not engineers. So the BRAIN Initiative supports technology, and it was crucial that we had the support. I will pause now. So again, you can email me for any of the reagents or if there's interest in the work, and I'll pause and take questions.
Thank you very much, Viviana, for that amazing talk, and we're open for questions.
Quick comments. Thanks so much, Viviana. I'm amazed by the amazing AAV vector you use. So I'm very happy to make some collaboration with us, because in ION we have huge amount of marmoset available. So very interesting to make like an autism model and make injection to knock out some genes brain. So I'm very happy to read the paper and see how we work together to make some new modeling work.
Thank you so much. I'm sure you'll make very good use of it, and the capsids are on Agene already, and I'm very happy to provide guidance on their use.
Great. Thank you so much.
Okay.
Hi, Viviana.
Hi.
Hi. Could you hear me?
Yes.
I'm really excited about the work, and according to your exciting work, it indicated that the initial pathogenesis of Parkinson's disease might be located in gut, right? Yeah. And I also heard that some other groups, for example, Jin Zhang group in China, they discovered that the initial pathogenesis might also be blood derived, because they discovered that the red blood cells will release a large amount of alpha-synuclein and finally impale the BBB. Yeah, so I'm really eager to hear your opinion about, do you think there is some overlapping mechanisms, between these two theories, the gut-derived or blood-derived? Because when you inoculated the alpha-synuclein into the gut, also alpha-synuclein also enters the blood, right?
Absolutely. So what your question implies is that an overload of alpha-synuclein to the brain, whether is directly from the brain or other paths, the gut, permeated or dysfunctional BBB, those can all be risk factors. So what's important to recognize, because of research over decades, is that PD is multifactorial. And I want to make sure I don't create the impression that I think the gut is the first and the origin. Now, there's equally possible that it can start from the brain in some patients and from the gut in others. And we know that toxin can cause PD, genetic factors can cause PD. So I think it's a multifactorial problem. And because of what we know now in terms of the immune influence on neurodegeneration, it is very likely that any perturbation in the blood-brain barrier could lead to factors, whether they're blood factors, right, into the brain, and that could maybe stress the lysosomal system, and that might not be able to cope with protein, with degraded proteins. So it's not, I think, one or the other. I think in terms of Parkinson patients alone, if we take them, we can create many groups and many causes for the risk factor. Not all of the patients will benefit from the same approach. Some might be very good candidates for deep brain stimulation. Some might be better candidates, for example, for immune blockers. So it would be very interesting to see how the area develops. But I think what we can say is that we can't afford the luxury of thinking about Parkinson as only a brain disorder.
Oh, it's really inspiring. So maybe the patients groups are heterogeneous. There is no universal mechanisms, right? Thank you. Thank you.
Thank you for the question.
Viviana, from your paper, it was exciting to see both the age dependent and then also the effects of GBA on alpha-synuclein spreading from the gut to the brain. I'm wondering if in later studies you guys have tried this in heterozygous alpha-synuclein mice or mice with a little bit less alpha-synuclein, and if that somehow delayed the spread. I'm just thinking if there's, knowing that it starts in the gut, if there's some ways of delaying the spread by targeting alpha-synuclein itself.
That's an excellent idea, Aaron. And this is one of the areas that we're hoping to pursue through the ASAP collaborative project by having a dial to dial down or up alpha-synuclein, and then see, depending on that dependency, see the timelines and what a reasonable timeline would be for genetic intervention. So I fully agree. And I think being aware of what level of overexpression would require intervention, that would be very important. So it's in the works. We are still at the point of developing the tools. And I think we can't make fast progress if we're locked in transgenics. So that's, yeah.
And it also might be of interest to see how LRRK2, mutant LRRK2 might converge on alpha-synuclein spreading from the gut.
For sure, the GBA delivery caused a partial rescue, and I want to emphasize that, leaving room for other pathways that are involved.
Got it. Okay. Other questions? Okay. This was a great crowd today and a great pair of talks. Thanks so much, Viviana. I think it also underscores that if you develop really powerful tools and you share them broadly like you have, they'll be very useful and that will increase the impact. So thanks so much for sharing all of this with us and joining. And thanks to everyone for tuning in. I don't see E-Rao here, but thank him for giving a great talk.
Okay, thanks,
see everyone next week.
See you all next week. Thank you.